We're collaborating with a wonderful group at Florida State, including Wilgenbusch, who's here from the Department of Scientific Computing, Gallivan, who's a professor of mathematics, and three math PhD students, Huang, Wen, and Melissa. Guifang and Wen and Jeremy are all here. And much of the specific results, to the extent that I'll talk about them today, are the result of their work. So they'd be happy to answer those questions if you have them. So I'm going to give mostly just a broad overview of the approach we're taking to addressing this problem. So I'm going to talk about some motivations, the network approaches that we're using to address this problem, some potential applications, sort of brainstorming session, if you will. I think there'll be many more than I talk about today. Just a brief mention of some initial results, and then some software that we've been working on to perform these kinds of tests. So the motivation comes from this notion of sets of trees. So in almost any phylogenetic analysis, we're going to end up with some large set of phylogenies, and these could represent different things. They often represent uncertainty in the estimate that comes from any individual gene tree, if it's a bootstrapping analysis or posterior distribution from a Bayesian analysis. They could also represent a series of point estimates from different genes across the genome, or to scale it up even bigger, they could represent a series of distributions of uncertainty from many different genes. So what we're interested in doing is asking, it's not useful often to report this entire set, so how do we efficiently and usefully summarize that set of trees? Now, we're certainly not the first ones to think about this, and perhaps the most commonly used solution to this problem is to present a consensus tree of some sort, a sort of average, if you will. So on this consensus tree, we label the bipartitions in that tree with the frequency with which we find those bipartitions in our initial tree set. There are alternative approaches, though. So some work by Karen and Bruce Ranalla has suggested the use of agreement subtrees. So this is a highly supported subtree that we find in many of our original input trees. So we can prune out some of the taxa until we find this subtree that agrees with many of the original input trees. There are also clustering-based approaches that Candy has worked on, she's here. And in this approach, we report multiple consensus trees, but we cluster the trees that go into each of those consensus trees to minimize the amount of information lost in the full distribution, while still representing the variation in that tree set. There are also approaches primarily to visualization involving dimensionality reduction. So we take highly dimensional tree space, and we project it into 2 or maybe 3 dimensions. So this was proposed by, I think, first by Hillis et al. In 2005. This provides a nice sort of visual summary of the tree space that we're looking at. So our approaches extend and complement, I think, these approaches in a lot of ways. So what we're interested in doing is using network-based approaches. So the first type of network we use is actually a network of topologies, or a network of trees. So the nodes in this network represent individual topologies or individual draws from some distribution. And the branches, or the edges in this network, represent the tree-to-tree affinities or similarities. So if you calculate the tree-to-tree distance, the edge weight here would represent something maybe like the inverse of that sort of distance. So this would represent a tree set where we have relatively uniform similarities among all the trees in this set. We could also, of course, have tree sets where we have a few much more similar topologies in sort of 2 distinct clusters. So we have low similarity edges that separate these clusters with high similarity edges in between them. Now, I think one thing that sets our approach apart maybe from some of the previous approaches is that we also leverage networks of bipartitions as well as individual trees. So each node in this network represents an individual bipartition, and again, we connect those nodes with edges. But we can't use similarity in this case because, of course, bipartitions are always distinct from one another. So instead, what we use are covariances of bipartitions. We can get a sense for what covariances might tell us about those bipartitions. If you think about this sort of consensus tree where we have 2 branches, an orange branch and a blue branch, and both are present in 51% of the input trees. Now, in this first sort of sampling, let's say 51% of the trees in our initial set had both the orange branch and the blue branch, and 49% had neither of those. Then the covariance between orange and blue and presence absence across that set would be 0.25. However, if orange and blue are sometimes associated with one another and sometimes not, and this is essentially So to give you a real example of what a network like this might look like, this is the type of bipartition covariance network that results from a uniform distribution of topologies. So here, just the structure of the trees themselves, of course, inhibits certain conflicting bipartitions from being in the same tree. So we have a lot of weak negative covariances, and to balance that out, we end up with some weak positive covariances. But we don't have a lot of sort of structure to this network. And we can contrast that with the kind of case where we might have two equally frequent topologies in our input set, which we get very strong positive covariances between the two branches. In this case, they're present in each of those trees, and then strong negative covariances between the branches that differ between those trees. And I'll come back to this idea a little bit later. So some things that we might be able to do with these networks. We can, of course, visualize them. And so anybody who's looked at sort of gene interactions has probably seen these sorts of plots endearingly termed hairballs. So we can get some sense for network structure by doing sort of like a dimensionality reduction sort of plot, and then just drawing edges between all the nodes in the network. And for small networks, this might work pretty well. For larger networks, we tend to lose a lot of information here, I think. Another approach to presenting the information contained in the network in a slightly more ordered way is to use this thing called a hive plot. And in a hive plot, you can essentially define an arbitrary number of axes, and you can define them in a variety of ways. So here I've defined an individual axis for 3 different genes, say, each of which contains a set of topologies. And then we can order, you can think about each of these axes as just being an ordered series of points. And so each of the nodes in our network is placed on this axis, and in this case, we're ordering them by the frequency with which we find that topology in each of those sets of input trees. And then we can do a variety of things like draw edges between those axes. So if, say, genes 1 and 2 are highly important in how they support those different topologies, we draw lines between the identical topologies from those 2 input tree sets. We see a lot of parallel lines going between genes 1 and 2. But you'll notice gene 3 here seems to have a very different, saying something very different about the relative rankings of those topologies. So high frequency topologies for gene 3 are low frequency for both genes 1 and 2. And there's a variety of ways that you can draw these lines to depict different sorts of information. We might also be able to use the structure of these networks to assess model fit in a parametric bootstrapping or a posterior prediction framework. So if we simulate data under a model we've used to analyze that data, we can compare the induced network structure in our empirical data. So if we did a bootstrapping analysis on our original empirical data, say, we end up with a network structure like this. We do a bootstrapping analysis on our simulated data, we end up with a network structure that looks like this consistently. This should suggest some disconnect between our empirical data and our simulated data, suggesting that our model isn't capturing something that's important in our empirical data. One thing that was probably obvious from the example networks that I showed before is that we can detect the presence of distinct phylogenetic signals in these networks. So if we look at a network like this, in this case it's sort of obvious that we have this cluster of trees down here and this cluster of trees up here. But there's a whole suite of formal methods that have been developed for detecting these kinds of communities within networks, community detection methods. And so we can use those methods to formally draw boundaries essentially between sets of topologies within that method. So it's an objective way to enumerate and identify distinct phylogenetic signals. We can also do that same sort of clustering or community detection on the network of bipartitions. So in this case, again, it's sort of a trivial example, but it becomes very obvious that we have 2 very distinct clusters. I should have pointed this out at the beginning, but one fundamental distinction between these bipartition networks and the topology networks is that you can have both positively and negatively weighted edges in these networks, but you can only have positively weighted edges in the topology networks. So we can apply different kinds of community detections on those 2 different kinds of networks. Once we've detected these communities, we can also plot other kinds of hive plots. So we could actually define 2 different kinds of axes. So we could have gene axes and community axes. We could ask how similar are the rankings of the topologies within a gene as compared to 2 different communities. So if those 2 genes have very distinct signals, we've done community detection on the whole set of input trees that gene 1 corresponds to community 1 very distinctly in this case, and gene 2 to community 2. But we could have more complicated cases where, say, gene 1 only supports the trees in community 1, but gene 2 has a mixture of support for trees in community 1 and community 2. And again, there's many other sort of formulations of these types of plots that we can use. So we've done some proof of principle simulations and analysis so far. So Jeremy Ash presented a poster last night that goes into this in a lot more detail. So you can talk to myself or to him or any of us afterwards if you'd like more detail on this. But just to give you some results, we did a simulation where we used two different input trees with differing topologies. And in particular, they differ just in the placement of one taxon, taxon 5. This is a sort of rogue taxon-like problem. The colored branches in these two topologies show the bipartitions that conflict across these two input trees. So we simulated separate data sets based on these two input trees. We combined them, did a bootstrapping analysis, and then we're exploring these sort of network methods for summarizing this bootstrap set. So if you just do a majority rule consensus for the entire set of trees that come out of this analysis, you'll actually place taxon 5 halfway in between the two input positions. And you'll notice there's low support sort of distributed throughout the tree. But it's not immediately clear from this single summary if that's just due to a lack of information or conflicting signal on exactly where taxon 5 might go to produce this sort of low support in these internal bipartitions. So if you create this is the actual bipartition covariance network from that analysis. So you'll notice we end up with a lot of sort of what we call free nodes, unconnected nodes. These are branches that are found in all of our input trees. So they have covariance of 0 with everything else. And then we have a lot of negative edges here that separate two distinct communities with positive edges between them. So it's pretty easy for community detection methods to separate these two out. And it turns out those sets of branches correspond exactly to the bipartitions that conflict between our two input trees. We do community detection using the topology networks. We end up with 3, and then in this case we just calculated consensus trees on the 3 distinct communities that come from those topology networks. You'll notice that we end up with 3 communities in this case, but we then colored the bipartitions by the communities from the bipartition networks. And you'll notice we get one tree that corresponds to one of our original input trees, one that corresponds to the other, and then one with this position of 5 in between the two original positions. So we can actually identify the bipartitions that come from each of our distinct input trees in this case. So this has been implemented in Treescaper, and Wen's going to give a lightning talk at IEGO Bio about Treescaper. So Treescaper is available. I, of course, forgot to put the URL here, but if you go to SourceForge and just search for Treescaper, you can find it. And Treescaper is already capable of calculating these networks and doing community detection and things like that. In the future, we'd like to implement this in a web-based interface. We've talked to some science gateways about making it available as a tool within some of these larger toolkits. So thank you very much for your attention, and I'd be happy to